Good afternoon, everyone. Welcome to the last session of this amazing conference, the Genomic Science in Cohorts session. My name is Ines Atmosukarto, and I am from the Indonesian Ministry of Health, and it's my pleasure to moderate this session, which aims to explore how the cohort infrastructure can be used as a source for genomic research. Now, before we start, I'll remind everyone that to please participate in the Q&A session by using Pigeonhole Live and log into Pigeonhole and key in password PIC24. I think everybody's quite familiar with this. As we have a hard finish at 5:20, right? I see them nodding. So we'll start with our first of three speakers, Professor Jian Jun Liu from Genome Institute of Singapore. I think Professor doesn't need much introduction, but he's currently the acting executive director and A-star distinguished fellow at the Genome Institute of Singapore and professor at the Yong Lu Lin School of Medicine at the National University of Singapore. I think I will ask you to take the floor. Thank you very much.
Thank you. Thank you for the introduction. Can you hear me? If yes, I don't need to use speaker. Well, otherwise, I think it's a bit better. Okay. Yes. Can we have eyes? Yes. For my talk, I'll focus on the one particular research effort within GS, trying to build a new reference genome for Indian population. So, what is a reference genome? Well, reference for definition, basically a sequencing database where presenting one of the typical human genomes, full sequence. That is called, a reference. But the functionality, they have three main applications. One, we all view them as a map. Every time you do a sequence, you need to know where the reads come from. You need this reference allow you to determine which part of the human genome this read come from. They call the mapping function. Then they also use it as a standard sequence function. Well, if you want to know whether a particular person carry out a particular variant, where you compare to? Well, you need a common ground. This will be called standard sequence at this moment. To allow you to see that, oh, this person carry a variant that are different from the standard sequencing. You use a way to do that. There's other function that catalog all the different functional units of the sequence of the genomes, all these things. There's probably less of a topic today. And then they have a widely application in many research. I do not want to really spend much time to talk about that. You probably should be familiar very much. So the first reference genome was January 2003 when the Human Genome Project was finished, the first draft. After that, there is a serious continuous effort to try to improve it. Because everybody knows there's errors, there's gaps, there's not complete. And so they always continue. At this moment, there are two major international efforts trying to get this better or more accurate reference. One is called the D2D or telomere to telomere. The aim of that one is trying to really get the full length of base pair, resolution, a complete sequence of human genome, end to end. Okay, and another one is actually just recognize the value called the diversity. Now people recognize that use a single reference genome as your reference to do the variant calling, sometimes not really complete. If you're using a multiple genome as a reference, we call the pan genome as reference, then you can improve your efficiency of variant calling, sometimes even mapping. So that's why there's ongoing two major international efforts, one called the T2D, one called the pan genome reference. Unfortunately, for both of these international efforts, the Asian population are well underrepresented. That's why we do need our own effort to create this reference for the Singapore's National Precision Medicine Program. So here it's just basically three major genomes that has been published, called the reference genome, one that everybody knows, MS13, one the HG002. Then there'll be Chinese genomes that published recently, right? And this is basically to show you what kind of data they are using to construct this reference. And the bottom is actually the genome we are currently working on. We're using trio structure, and we build the reference genome use the child DNA. And the reason we use trio, because they will help us to phase the hepatide or the chromosome more accurately. Okay? And then you can see what kind of data we are generating to do this. Then we'll cut, very simple. We're using two different commonly used assembler. One is Hyphaesium, one is Vercal. You can sample, all the genome. You can see all this presentation of different chromosomes. Some of them, you get kind of a whole genome contig, but very often, most time, they're not. They're basically multiple contigs and instead of a complete. Then in that case, you need to link them up. We call the scaffold. Have to link it or called contig into the chromosome level. We call the scaffolds. How to do that? Well, we use two methods. One is basically using high-C or omic-C data, because this is show you what is the sequence relative position along the genome. They can do that, but not always resolve everything. We still have a few, contig cannot be reliable or convincingly determine their relative locations or relative positions. Then we take CM13 as a reference, just to line them up. And then basically, so when you line them up, then you have to, how do you create this chromosome scaffold? Well, there's a gap, because when the two contig, next to each other, but you don't know what is in between them, so we just put all the N there and use artificially 500 N's there to link them up. Okay, that's how we generate these chromosome level scaffolds. After that, then you're just wondering, well, is this a gap? How do we close it? The first question we ask, is it real gap or not real gap? Sometimes the two contigs supposed to be linked together. For whatever reason we don't fully understand, the assembler has not been connecting them. In that situation, we call the artificial gap. No gap, we just link them up. Sometimes they do call the real gap, meaning that there is a sequencing in the middle between two contigs. The sequence is lost during the assembling process. We need to bring back the sequence. So the first step to do the determine what the real gap, what is not real gap. To do that, very simple. You just remove all the gap, you remap all the reads to this sampling. Okay? If there's no gap, well, just remove it, create a perfect reference. Then all the reads can be perfectly mapped to it. But if the real gap, if you remove them, you short something. When you map the reads against that, well, you can see variations, because even though the sampling comes from these reads, but because you artificially remove something, you create the variation, most likely insertion. Well, the reads have something you're not reflected in your reference. That's where the insertion comes in. So that's what allows to determine which gap is real, which gap is not real. If not real, we just link them up. If real, then this example, what it means, artificial gap. There's a gap there. You remove everything mapped perfectly. So I would cut short. How do we fill the gap? The real gap. We know the sequence is missing. Right? And for that, we use basically two different approaches. The first one is kind of automatic approach called the GGS algorithm. What they do is just looking for all the reads in your data, try to see whether they have found any read that can span in the whole gap. There's on one end on the one contig, another end, another contig, trying to use one to guess where is the sequence in between the two contig look like. But that is only go very few, because if you have a lot of these reads, then they should be merged already. That's why you only have a very few situation where maybe there's issue with the reads quality or number of reads, they will not be naturally linked. But if solve a couple, but most of these cannot do that. So then most of them will have to use in other way, many different way to close in all this gap. Particularly because we have a lot of other type data, duplex data from the ONTs and different assemblers. Basically, we do regional resampling again. The gap where we think there is a gap, real gap, we retrieve all the reads and redo the sampling and look at all this newly assembled contig to see which one can bridge the gap. By that, you'll be able to close the gap. That's how we did. So we close all the gap by this way. So now we really have a so-called chromosome level scaffolds. But after that, well, there's going to be errors. We understand. So a bit about polishing it. This is a very, very tedious process. Try to polish your assembly to make them as accurate as possible. I think we never end of it, right? Which is as much as you can do.
There's two steps we do it. We do the automatic one, because that's basically redo the mapping. If your reads, there's no error, your reads map to your assembly, you should have no variation, right? Because your assembly perfectly supported by your reads. But if there is a variation come out, there are indication, well, probably something wrong with your assembly. If all the reads tell you a homozygous, variance call come out, then you know your assembly have something wrong. You should follow the reads, change your reference. So that's called the automatic process. They can push, the quality score from 55 to 72. Basically, it's dramatic. Use this simple polishing, automatic polishing procedure, you can dramatically improve the quality. But after that, then you have for those who have 600 still error there. We can still see error. The automatic one cannot fix it because why? Because even the read cell cannot agree to each other. That does not have sufficient information IOs to fix it automatically. Then you have to do, we call the manual creation. People have to go in, look at all the reads, the IGV files to manually, painfully crap. We basically have two groups here, my group and the middle group. We assign all the 600 things to each individual. You gotta go and do it in 2 weeks' time. You look at each one of them. Try to do as much correction you can do, within reasonable time. Otherwise, you'll never end with. So we think we do the reasonable thing. We basically, up today, we kind of resolve about half of it. 339 error has been fixed, but it's still many. But definitely help us to further improve the quality. But now it's reached the 74 and the 72. Telomere. Are you sure you are complete? Without telomere, you cannot call yourself complete. That's why I call telomere to telomere, reference. So that's why we look at the result. We notice that, well, only partial chromosome you can see the telomere. Many of them not there. Well, that's a problem. So then we says, okay, now we need to pull all the telomere-related reads out of our data. How to do that? With our reference does not have a telomere, so you cannot use our own reference to do that. So we just take all the telomere sequences from published reference genome and then use it as a way to fish out all the telomere-related reads. There's a lot of say why you need to know more everything you can have there because diversity there. You need all the reference to be able to ensure you can maximum retrieve all the telomere-related reads from your data. After get that, well, you do the local, consensus kind of assembly. Then after you generate this, you patch back to your end. That's why we managed to generate. We figure out all the called the telomere to telomere genomes. We done that. Good. So then the RDNA, this is the devils. Everybody understand, you do the, general human reference genomes, the 5, RDNA regions, the 10 clusters, 5, maternal. This is the devils. It's very, very hard to challenge. We still try to resolve it. At this moment, we roughly estimated how much of copies of RDNA in this particular Indian genomes, and we use a reads-based mapping approach to estimate relative numbers among these 10 clusters. So that's far. And at this moment, it's an active effort trying to see whether we can create a real RDNA sequence. If you think CM13 is a real sequence, no, not yet, definitely not real. Because what they do is actually they define all the RDNA sequences, line them up to define the morphs, and use them more to create artificial. You have 5 copies. I have 5 morphs. I just stick them together and insert into the genome. I got the RD, but that is not real. Nobody knows whether it's real sequence, but it's reflecting the nature of the repeat they carry and the copy number. But in terms of orders, in terms of which way organize them, nobody knows. There's also so. This is probably mostly likely the outcome we're going to end up, but we're trying to, for some of the small part of that, let's see whether we can get real sequence out. That's what we'll do right now. So that's basically up to date. So we are getting the, paternal, maternal, because we have the, parental allows to separate them out, determine them, and we know they're all telomere to telomere, and we know the quality score. So you do the comparison, there's assembly evaluation, everything perfect, collect, assemble, there's no switch errors, whatever. And the quality size, you can see the top 2 is our genomes. The rest is basically the published reference genomes. In terms of length, qualities, we up there. For this, we are one of the top. Very good, very good. And we did kind of consistency checking, right? You do the pairwise comparison with the CM30 to see where is a major difference. Well, good thing is no major difference, but there are certain differences, but all around the centromere region. Okay, you do see that centromere region. That's why we take a look at centromeres. Oh, is something terrible wrong or what? Yeah. So first of all, see, do we have all the sequence there? You rather look like there's no kind of major drop in the particular region of the centromere so that we got all the sequence there. And we check all the repeat units or repeat sequence they carry, right? They call this alpha-satellites or the sequencing. You compare this with with the reference, CSR13 to see which unit we also see it, which one we don't see it. In terms of length, someone is longer than the CERM13, someone in the shorter. On the right side is a panel to show the relative length of the centromere between the maternal and the paternal chromosome. And you can see some of them is longer than one side or shorter than the other side. And we do see a few repeat units there. It's called the plus, show that is it some kind of unique sequencing in the Indian genome you have not seen in the CM13. Yes, we know there's a difference between the maternal. This is really demonstration how the centromere sequence look like for each of this chromosome between the paternal on the left side, maternal on the right side. You do see some of this chromosome, like a chromosome 19. The length is tremendously different. Well, I just learned this morning from Kai says that difference has biological meaning, have an impact during the meiosis or mitosis, between pairing. There is impact for that. So it's interesting to see there's such big diversity between the maternal, right? Well, the final thing I say is better to check, are we really looking at the Indian genome or not? Because in the lab, there's a lot of samples. What if someone submit the DNA sample, wrongly? When we are analyzing the Indian genome and have a Chinese genome, that would be a disaster, right? So that's why it's sanity check to see whether this is really Indian genome. Yes, true. It is a truly Indian genome. Good. Otherwise, I'll be the guy. So this is only the first genome going to cook out under the NPM program called the Asian Reference Genome Project. So in that project, we'll have a tier 2 project to generate the T2T reference for Chinese, Malay, Indian, each one of those. Then there'll be a more pan-genome reference later on to be generated for 100 genomes with at least 30 per acidical groups. So I can show you what kind of a planned data generation, but this is basically the nutshell of a description of what the Asian Reference Genome Project in Singapore look like. There are many other efforts. We do hope have opportunity to collaborate with people, learning from each other, whoever work on the Pan-Genome Project, particularly China, Shuhua's in the morning there. Sure. Yes, this is my acknowledgement, and this is teams, my group, and Melis group. And particularly, I would like to acknowledge William Huang from National Cancer Center. Very long time ago, he's made a major effort collecting all the trails. This is one of the trails we're using to generate this Indian genome reference. Thank you very much.
Thank you very much. What an amazing start to this last session. I will now go on and invite Assistant Professor Max Lam to present to us his talk. Max is the Deputy Chief Scientific Officer of PRECISE, which is, we all know, the central entity established to implement phase 2 of Singapore's National Precision Medicine Strategy. I think Dr. Lam's interest is mostly in cognitive and psychiatric genetics, and I'm sure we'll hear about that now. Thank you. Floor's to you.
All right, good afternoon, everyone, and thank you for staying with us until this final session. So today, I'm excited to introduce you to our research efforts in cognitive genetics and how we are utilizing and benefiting from large-scale community resources from biobanks and cohorts. So you might have noticed that my presentation is titled Cognitive Health rather than just Cognitive Genetics. And this is intentional and speaks to our aim to integrate cognitive and genomic resources that have been generated by our colleagues with the broader context of bringing together the mental and physical health research to uncover new insights, not only to advance our understanding of cognitive function, but contribute to the overall health and well-being of individuals. Right? So, why study cognitive genetics or cognitive health? The drive to explore this field stems from our deep-seated efforts to unravel the biological mechanisms that underpin psychiatric illnesses. Cognitive impairments are not just common in psychiatric disorders. They often play a significant role in the functional disability that patients experience. These cognitive deficits directly impact our patients' daily lives and overall quality of life, making it crucial for us to understand them a little bit more deeply. Now, over the past decade, the formation of large international consortia has accelerated the progress in this field, leading to remarkable advances in our understanding of the genetic architecture of psychiatric disorders. Now, critical studies by our group and others studying schizophrenia, bipolar disorder, and MDD have been instrumental in uncovering the genetic associations with psychiatric conditions. Now, despite these breakthroughs, our field continues to face significant challenges, particularly when it comes to translating these genetic findings into clinical practice. Cognitive genetics offers a unique lens through which we can explore how genetic factors influence not only psychiatric disorders, but also the cognitive and functional deficits that are so closely intertwined with these conditions. Now, one of the central concepts in cognitive genetics is Spearman's G, a factor that represents general cognitive ability. Spearman's G is not just a single measure, but an underlying construct that emerges as the intersection across various cognitive domains, such as memory, reasoning, spatial ability, and verbal skills. This construct is remarkably consistent across different cognitive tasks, making it a robust and reliable phenotype for genetic studies. In our research, cognitive task performance is operationalized through Spearman's G, serving as a key phenotype that allows us to study the genetic factors influencing overall cognitive function. By focusing on Spearman's G, we ensure that our findings are relevant across a wide array of cognitive tests. This approach provides a comprehensive view of cognitive ability, as G is consistently found to be the primary factor when we run the first principal component, or extract the first principal component from diverse cognitive or neuropsychological tests. The consistency reinforces the idea that G captures general cognitive capacity that's applicable across different contexts and tasks, making it an invaluable tool for cognitive genetics research. Cognitive function is a critical area of study within complex trait genomics, extending far beyond just psychiatric research, and it serves as a key trait for understanding a wide range of behavioral and psychological processes. Cognitive performance is not only a well-powered trait for large-scale genetic investigations, but also increasingly measured in extensive biobank-level studies. And the cognitive tests remain, today, the most reliable and direct measure of brain function, making cognitive performance quite an ideal candidate for genetic research. And furthermore, the advanced methodologies and well-established statistical genetics approach that have been developed over the years do allow us to explore the genetic architecture of cognitive performance with reasonable precision and accuracy. Now, if you look at a figure on the slide, the y-axis shows the number of new genomic loci discovered, and the x-axis, the studies that have been published over time. And you can see from this figure, we are currently entering into an exciting phase where genetic studies on cognitive function are yielding rapid and significant returns. Over the past 8 years, our team has been at the forefront of this effort, contributing to the uncovering of key genetic variants and understanding of how these variants influence cognitive abilities. Certainly, these numbers are nowhere near genomic studies that have reported BMI and height, where they're reporting millions and millions of individuals. Nonetheless, we hope that our efforts are not only advancing our knowledge of cognitive genetics, but also paving the way to identify specific genetic factors that can be targeted in therapeutic interventions. Now, in this section, I'd like to provide an overview of some of our recent research highlighting that from our group, and our work has primarily focused on pleiotropy analysis, particularly investigating relationships between schizophrenia, education attainment, cognitive task performance. In addition, we have explored proteomic profiles using Mendelian randomization to understand the links between cognitive traits and psychopathology. Beyond these efforts, we have been extending our research into Asian populations to ensure that our findings are broadly applicable and inclusive. So a key component of this expansion is the Precise SG100K program, which presents a unique opportunity to broaden cognitive health research. And by leveraging on the extensive cohort that Precise brings, we can explore cognitive function and its genetic underpinnings in a more diverse population study. So let me explain a little bit about pleiotropy. So pleiotropy, or the phenomenon where a key gene influences multiple traits, is a key concept in genetic studies, particularly in psychiatry. So understanding pleiotropy is crucial because it helps us uncover how genetic factors contribute to various aspects of cognitive function and psychopathology. Now, let me try to describe this figure on the slide. It illustrates here a gene contributing to both a quantitative trait, and in our case, cognitive function, and a liability threshold for conditions like major depressive disorder, MDD, a cardiovascular disease. This visual representation highlights the dual role a gene can play in influencing both a continuous trait and a risk threshold for developing a disease. It is important to note that these traits, however, when it comes to psychiatric illness and cognitive functions, are polygenic, meaning that it's not only just one gene driving this effect, but rather multiple genes, each contributing to small effects. The interplay of these multiple genetic factors form a complex network that underlies both cognitive function and disease susceptibility. So by studying pleiotropic effects, we can gain deeper insights into the shared genetic architecture of these complex traits, helping us to better understand the genetic basis of cognitive health and disease. And that's what we've begun to do, looking into the pleiotropic patterns of cognitive function and psychopathology that you see on the slide here. So in this study, we demonstrated that indeed, broad pleiotropy exists across a myriad of psychiatric conditions with cognitive function. So to investigate pleiotropic relationships, we use several advanced statistical genetics approaches, and these methods allow us to identify genetic variants that are associated across multiple traits, as I've mentioned earlier. One of the major outcomes of our research has been the identification of shared genomic loci between cognitive dimensions and psychopathology. And these shared loci will help us understand the biological mechanisms that drive cognitive function and its relationship with psychiatric disorder. And more importantly, we hope that this research has the potential to pave the way for personalized medicine. So in this study, we began by bringing together data from large-scale biobanks, genetic studies, and so those are the summary statistics that we can get from these resources. So after quality control, we looked into variant association patterns across the outcome variables, which we could then categorize as concordant or discordant based on the genetic associations. Concordant loci or regions, for instance, are where the genetic variants have similar effects on both cognitive function and psychiatric disorders, indicating a shared genetic influence. On the other hand, discordant loci exhibit opposing effects on these traits, suggesting that some genetic factors might contribute to cognitive function while simultaneously increase the risk for psychiatric conditions. So to dig deeper into the underlying biology of these associations, we conducted follow-up biological annotations, gene set analysis, including running brain span analysis to examine gene expression profiles in postmortem brain tissue, helping us understand a little bit better how these genetic associations contribute to cognitive abilities and psychiatric disorders. So here is the Manhattan plot for running this pleiotropy analysis. And pleiotropy analysis do play quite a crucial role in enhancing genetic discovery, as you can see here, especially when we are dealing with traits that are known to be kind of related to each other. By leveraging pleiotropic relationships, we can uncover genetic variants that might otherwise remain hidden when studying traits in isolation. So this approach has proven to be quite fruitful, where we identified a total of 768 genomic loci that are significant, associated jointly with cognition, schizophrenia, and education attainment, including 166 of them that are novel and hasn't been previously reported. Now, what's particularly exciting is we were not only able to identify novel loci, but this analysis is giving us a new category of loci, which we call dual loci. And these are regions of the genome where both concordant and discordant variants are located in close proximity. The identification of dual loci is particularly intriguing because it suggests complex interplay of genetic influences within these regions and potentially offering some new insights into biological mechanisms that drive both the cognitive function and psychiatric conditions. So to deeper understand the genes underlying each category of loci, concordant, discordant, and dual, we conducted gene set analysis, and this analysis allowed us to identify broad biological processes associated with these loci. So specifically, we found that concordant loci were linked to neurodevelopmental processes, discordant loci linked to synaptic regulation, and the dual loci appeared to involve translational factors. We then sought to validate these findings using an independent data set comprising postmortem brain expression data from the BrainSpan project. And the results from this analysis broadly support the gene set annotations, reinforcing our initial conclusions about the biological roles of these loci. And what's particularly interesting here is the observation that the dual loci seem to be tied to broader neurodegenerative component and suggest that these regions of the genome may play a significant role in degenerative processes and could be crucial to opening up opportunities for follow-up subsequent studies. Right? So I'd like to change gears a little bit to talk about Mendelian randomization, and that's an incredibly powerful tool in our arsenal, particularly when large-scale resources that has been made available through UK Biobank. And the UK Biobank has recently published the proteomic profiles over 50,000 individuals, providing an unprecedented resource for researchers. And this data set opened up the exciting possibility of investigating whether specific proteins in the blood are related to cognitive and mental health traits. Now, with these resources at our disposal, we can now ask the critical question about the relationship between proteins and psychiatric or cognitive condition. The ambition here is significant. So by leveraging MR, we hope that we can one day identify blood biomarkers that could serve as early indicators of psychiatric disorders. The MR analysis allows us to establish causal relationships by using genetic variants such to effectively help us infer causal effects of the exposure, such as protein levels on outcomes like mental health or cognitive traits. Now, with interest in time, I'm not going to go into too much details on the methodology, and I encourage the audience to check out the preprint listed here. But I just want to make the point that we have identified several protein signatures associated with cognitive function and psychopathology. Many of these proteins are related to immune function, such as interleukins, complement factors, and our findings also shed light on proteins involving the regulation of inflammatory responses, which may play a critical role in cognitive function and mental health. And these discoveries, we feel, provide valuable insights into the shared etiology of psychiatric conditions and cognitive ability. As part of our commitment to increasing the diversity of genetic research, we are actively working to expand cognitive genomics studies into Asian populations, bringing together large-scale genomic data generated by the Taiwan Biobank, Korean Biobank, China Kadoorie Biobank, as well as other groups that have generated discovery GWAS in schizophrenia, education, and major depressive disorder. Much of the research in cognitive genetics that I have described so far has been heavily focused on Eurocentric populations, leaving a significant gap in our understanding of how these findings might apply across diverse ancestry. So here, we are starting by applying pleiotropic methods that I described earlier to a proxy phenotype for cognitive function, education, attainment, which serves as an important initial step to understand cognitive genetics within the Asian population. So this effort led us to discover 32 novel regions in the genome associated with depression and schizophrenia in Asian ancestry populations, underscoring the critical importance of diverse representation in genetic research. And some of these novel loci have not been reported in European-based or GWASs of these traits. What's interesting is when we take these results and put them into polygenic risk score prediction, we do see that bringing together these pleiotropically generated summary statistics or shared genetic architecture across the trait is that we can refine our predictions and better identify individuals who are most at risk of developing psychiatric disorders, looking into the genetics of that are intersecting between psychiatric and cognitive function. And we think that this might be indexing putative biology that might be critical for polygenic risk score prediction. Now, you might ask, well, this is still education attainment as a proxy to cognitive function. Can we measure cognition more directly? So here, I'd like to introduce the PRECISE SG100K Cognitive and Mental Health Flagship Program, which represents the largest cognitive data collection effort within the Asian cohort at this point. This program includes comprehensive data from 100,000 Singaporean. The 50,000 release is imminent, and this research is embedded within the broader PRECISE SG100K initiative. The significance of this program lies in its potential to advance genomic diversity in cognitive health research, and we hope that would enable robust studies on genetic and environmental determinants of cognitive health in Asian populations. So over the next few years, we also plan to be to be generating community resource in these large-scale studies, not limited to just cognition, but more broadly. So here, I'm showing the overview of the phenotyping work within the Helios SG100K study and the cognitive measurements that we are curating. We do have a QR code where you can take a quick read of the preprint. So as part of the effort to initially understand cognitive ability here in Singapore, which we feel are broadly representative of an Asian population, we can carry out epidemiologically a phenome-wide association to see, at least at this initial phase, how cognitive function varies with other health measures here in Singapore. And on the slide, you will see the phenotype categories that we have compared cognitive function with. So expectedly, there are strong social environmental factors that are associated with cognitive function. But from a physical health perspective, we are also observing associations with waist-hip ratio, white blood cell counts, underscoring the importance of cognitive functions as a key phenotype in genetic research. So with that, I'd like to take this opportunity to thank all our collaborators, funding agencies, and institutions who have made this research possible.
Thank you, Max. What an amazing insight into the power of these large biobank data sets. Now, I would like to welcome Dr. Nicolas Bertin, who's with the Agency for Science, Technology and Research. Nicolas is currently leading the Genome Institute of Singapore's Genome Research Informatics and Data Science platform, is in charge of the population-scale genomic data management, governance and analysis for the PRECISE-led National Precision Medicine Program. Nicolas, floor is yours.
Thanks, Ines. All right. Good afternoon, everyone. And so what I will be presenting in the next 20 minutes is our first effort toward generating a comprehensive catalog of structural variation across ancestrally diverse Asian genome as part of the PRECISE effort. All right, so key motivation for why sort of like studying structural variation is the fact that like there's even structural variation actually in number of base pair they involve in the genome sort of is greater than that of like single base pair and like SNP. And so to some measure, they are actually structural variation are the main driver of a genetic variation in genomes. And so akin single nucleotide polymorphism, they do impact health and wellness, and there are some notable example, for example, a very famous structural variation in the BEAM genes that provide resistance to tyrosine kinase, and then like single nucleotide phenotype, pattern of structural variation and a prevalence vary across ancestries. And it just so happened, akin, like really the large a genetic variation catalog that Asian population are underrepresented in currently available global catalog of genetic variation, the like of like a Nomad 1000 Genome, CCDG, and then all those large international efforts. And so what's known of structural variation, natural structural variation occurring in healthy individual is largely a Eurocentric view, and so efforts are needed to like fill the gap and provide a more global and less biased overview of structural variation. All the more that like Asian population actually account for the vast majority of the population worldwide, and so what we did is really leverage on the Singapore National Precision Medicine program to really like undergo and fill up this gap in sort of trying to catalog structural variation at a large scale. And so we took advantage of like phase 1 of the PRECISE National Precision Medicine program, which basically provided whole genome sequencing from 10,000 participants, short read whole genome sequencing, a mix of like 15x, 30x, which I will discuss, had some implication in our ability to generate this catalog. But more importantly, this small number, just 10,000 participants, already was contributing more than all existing efforts at generating reference catalog for structural variation. So to a large extent, because of the nature of the data we had at hand, which is like short read sequencing, some library even at like 15x depth sequencing, we had to sort of, despite the vast diversity of like events that lead to structural variation, had to purposefully decide on like limiting ourselves to like the easier to detect with such technology type of structural variation events, namely deletion, insertion, and duplication event. Sort of leaving aside much more complex translocation event and massive inversion, for which short read sequencing quickly shows some limitation. And so to define to a reasonable extent and characterize structural variation, what we did is try to find sort of like define a workflow that would allow us to like robustly detect structural variation in individuals, but also like have a concise catalog across individual, and then basically joint calling variant across the entire 10,000 NPM phase 1 collection. And so to establish this particular detection algorithm, it was very important for us to like benchmark it. And for that, what we've done is we've taken advantage of the 1,000 genome program, which had the very good fortune to not only have like very deep sequencing of 3,200 about sample, but also for a subset of them very early on, like whole genome sequencing using long reads. And then we took advantage of the work from like Ebert et al., who had really established like structural variation. And then we used that as a ground truth against which we could sort of design our own detection algorithm and pipeline, and sort of like pick and choose what were like the best algorithm combination thereof to get a robust pipeline for asserting structural variation in Chinese, Indian, and Malay. And so this is sort of like a quick overview of sort of like the results, like like what's the like precision of the pipeline, and more importantly the F1 score, which provides sort of a global overview, which led us to actually like against all odds, instead of sort of combining many algorithms that were affiliated, to actually choose like Menta only when it came to asserting insertion and deletion only. Also doing this particular benchmarking effort allowed us to sort of like gauge what we were losing by having just 15x sequence short read sequencing, as opposed to the more commonly used now and quite standard 30x sequencing, and establish sort of like how much was lost in this effort. Also, to complement this approach, we had in-house a group developing algorithm specifically dedicated to robustly detecting duplication, and so Servindel, and we sort of also leverage on this particular algorithm to complement our duplication analysis, and in particular, like for tandem repeats, a region and sequence. And so all together, this created like the workflow you can see sort of like in this diagram, where we've basically combined so Menta's femur graph typer, Servindel 2, but also we leverage on MELT, which is an algorithm that is dedicated to identifying mobile elements in insertion type of event. So what we then did, having established a catalog of structural variation in 10,000 sample, is go about sort of like asserting sort of like what was the different characteristics of this particular catalog. And so unsurprisingly, the vast majority of the structural variation tend to be rare events. Also, the very large majority, about 70% of the events that we detected in those 10,000 participants were actually unique and never reported before in any other catalog. And so this like provided us with like a grand total of 75,000 robustly identified structural variation across 5,000 individuals. We also looked at the distribution of events per genome. Not surprisingly, so no notable difference in the number of deletion, insertion, or duplication across Chinese, Malay, or Indian individuals. But having the same number of events across genome does not mean those are necessarily the same events. And what we could see is like the pattern of a structural variation across the different and Ancestry actually recapitulate what can already be found in terms of like population structure from a principal component analysis when compared to single nucleotide polymorphism. What we then saw is that like not only was the like sort of vast majority of the events we saw were novel, but it was sort of, yeah, we basically, with this effort, enriched to a great extent the cataloging of structural variation, especially present in Chinese, Indian, and Malay population throughout the deletion, duplication, and insertion. Looking at the length distribution of those various events, we find like what was expected of like the different, like several modes, highlighting sort of the preponderance and the prevalence of transposable element, ALU line and SVA, in being major contributor to the structural variation across genomes. And so while the vast majority of the structural variation tended to be so like on the smaller size, we did identify in healthy individual or otherwise healthy at the point of blood collection in individual several very large deletion and duplication events. Yeah. What we look at also is like where were located those particular structural variant, insertion or deletion, along sort of like functional element of the genome. Unsurprisingly, we found that like structural variation tended to be like depleted across a genetic element, UTR and coding sequences, and also that like the common deletion, the one present at more than 1% in the general population, tended to be depleted in putative regulatory element, an answer and insulator. Common duplication, however, were enriched at a non-promoter H3K4 metal 3 region. We then look at 4 structural variation affecting a protein coding genes, the extent to which those structural variation would result in copy gain or loss of function, and identify that like about 3% of the catalog actually disrupt protein coding and lead to like complete or partial a loss of function. Looking particularly at genes that are known to be clinically relevant, we focused on the ACMG secondary finding lists and identified a 14 structural variants event affecting the coding sequence integrity in 11 ACMG gene. And then what I show here is one particular example in the TRDN gene, which is like a key component of the calcium release complex that we found in 3 otherwise healthy Chinese individual. And the key thing is really that gene is involved and associated with cardiac dysfunction and cardiac complication. Yeah. We also sort of looked into the potential of like ancestry specific, either like Indian, Chinese, or Malay structural variant, and like structural variation presents like predominantly with higher prevalence, like depending on the ethnic background, and sort of identified a grand total of like 10,000 structural variants that were actually more prevalent in Chinese, Indian or Malay. 86 of them actually sort of harbor, like were structural variation that affected a coding genes, 40 deletion, 44 duplication, and 2 insertions, yeah, that are both ancestry specific and would affect the biology of known genes. And so notable example of like known Asian-specific structural variants like involve like few of the genes highlighted here, STL3 and receptor for activated protein C, which were known to be more prevalent in Malay than Chinese or Indian. But we also find sort of like novel structural variants in like affecting a genes at a higher prevalence in like Indian sometimes and Chinese or Malay. One of the things also that we set out to do is to look at the linkage equilibrium between structural variant and SNP, with sort of like the hypothesis that many of lead GWAS SNP that fall into like intergenic region or intron may not be the causative variant for like a particular trait, but actually a structural variant which by virtue of how this particular GWAS has been done, like a genotyping array, for example, could not have been sort of like found and detected, yeah. And in doing so, that is like taking known GWAS catalog lead SNP, looking at a structural variant in strong linkage disequilibrium with those lead SNP, we could sort of like start making hypotheses about what structural variant might actually be the causative element. And then we identified a grand total of like about 400 structural variants that could be the potential a causative genetic event for the particular trait. And here is one example of like a loss of function overlapping like a loci, which is a much stronger case for the effect on psoriasis in East Asian ancestry individual. And so this work, which actually is the great work of like a talented postdoc and RO in my team, is currently under review in NatureCom, and then you can sort of have a look at the preprint already. And so future prospect actually is to like fully take advantage of the precise SG100K catalog and overcome some of the limitation and suffering Joanna had to go through to establish the SG10 catalog. That is, everything will be 30x deep sequence, no 15x library. We'll also have a much richer catalog of phenotypic information, like i.e. the slide that like Max showed earlier, like a gigantic effort from the Helios and NUS team, which will allow us to directly go after genome-wide association study. Also tied to the work presented by JJ earlier, we'll fully take advantage of the long-read project, because decisively, long-read is like the technology of choice when going after like structural variation, much more so than short-read. Yeah. And so, yeah, with that, I thank you all for your attention, and I'm like happy to take on questions.
Thank you very much, Nicolas. And I would like to call on Prof Liu, and I think we've lost Max. Max ran away before we could ask him questions. But maybe we can start with the questions for the other speakers first, and then we can bombard Max at the end. Amazing talks. Thank you very much for a really interesting last session. I'll start with JJ. How long did it take to fully resolve the 3 genome, including the manual curation? Is it feasible to do it for a large cohort study?
Okay, well, it take a long time. Particularly the manual curation part take a long time. There's, like I said, you probably know end of it. You can always spend more time to work out the last piece for that single puzzle, and it's always there. So I think realistically, after we done the first genome, we know where the bottleneck. And if we want to do large scale, we just have to compromise on some things. We have to see that the certain things will be not part of result because we cannot resolve them quickly. And then for other part, I think we can do much quickly because we have all the, at least some kind of automated or semi-automated pipeline to be handle it. Then what you need is basically access to large computing facilities, so that will be able to speed up the whole process. Yeah, feasible. Yeah, to do large scale.
Thank you very much. I'll move on to a question to Nicolas. Is there going to be a comparison for short read sequencing and long reads, or a reflex method to sequence long reads for genomes with interesting SVs?
So a comparison between like short read and long reads, I think there's no need. We know who's going to be the winner. That's quite obvious. The advantage of short read might just be in like the cost per genome, and so the ability with this approach to actually like sequence like twice more in individual than with long read, like I mean just like the cost ratio, and so like no comparison will be using both like as much data available as exists. Yeah.
So,
but this, I don't support that yet, because for the long read, we only sequence 10,000 out of 100,000, right? If you can help do the comparison, at least we know for the NGS-based SV calling, what is missing? What could be wrong? And at least because for the remaining 100,000, you still have a 90,000 genome that does not have a long read data. And then people will you're probably going to call the SV, or at least the NPM would like someone to call the SV out for this data sets. But with this comparison, at least we can sign certain annotation says in certain type of the calling result, be cautious because the error rate will be high. You're missing a lot of things. You only get a very, very, very partial picture. But for certain kind of structural variation, the maybe NGS may be quite good and reliable, so that you can really use it in the 100,000 genomes data. I think that is probably the comparison can contribute to this question. Yeah?
Could I just follow up? I'm from Indonesia. Indonesia is just starting its genome initiatives, and I was wondering if I could get some feedback from you on, how much effort should a new initiative with limited funding spend on long read sequencing? Is it something that we should focus on? And if so, what percentage maybe of the data? I was wondering if you could, give us your thoughts.
I can give my penny of thought, right? And well, if long read will be definitely good, give you much more comprehensive information, including both the invariation information and the methylation information, or or the modification information, but it's very expensive, right? And if your primary interest is only the SNPs or a few indels, the small indels, then NGS is very, very, very cost effective. So there's really no obvious answer. It's really depend what you want to achieve. Particularly for some program get started, you need to figure out what is your initial deliverable. They were able to convince government or the funding agency, allow you to go further. That's critical. You cannot go just one step to finish everything. You need to be very strategically think about it. What is the initial result that be interesting enough for all the stakeholders to be convinced, willing to support you to go further? I think if you define that question well, then you think what is the best way to deliver. Then you think you will find the right approach. It's a very, yeah.
Thank you. Nicolas?
Yeah. I mean, I completely agree with JJ. I mean, the obvious is really like technology keep on evolving. And so this is, the key question is more what is sort of like the right balance of like cost effectiveness, and then what you're after at the point where you're in a position to like start the program. And you can always wait tomorrow. There will always be a better technology, more cost-effective, but then this just delay the results and like obtaining outcome and being able to like make progress on sort of bringing the genomic information as like a key tool, a key measurement to improving health and wellness. Yeah. So.
Yeah, thank you. I guess it's finding that low-hanging fruit that's going to help us convince the funders. Max, thanks. I have a question for you here. What are your thoughts on trials for semaglutide in neural disorders, given that CVD impacts cognitive abilities and genes linked to CVD also affect cognition? And
it's a long question.
That's a long question, which I'm going to try and put up for you.
Okay.
Right. Can you see it on the tray?
Yeah. Yes. Okay. So I must say that I'm not so sure what's semaglutide. Can the person who asked the question briefly?
Well, put it this way: Max is not a doctor or clinician, and even don't know what that means. This drug or target.
Thank you for the microphone. So because it also come up in the earlier talks as well, semaglutide is basically Ozempic that helps people with the weight loss. And then it was mentioned that the genes that are coming up in the cardiovascular disease also very related to the cognitive genes. So that now, since you are trying to look for genes that is impactful for the cognitive abilities, will you also start getting hits that's actually impactful for the, metabolites, the body weight, insulin resistance? So that was the question.
The semaglutide, what is that? It's a compound or it's compound, right? Okay, yeah.
Yep.
It's targeting particular gene or target, do you know? Oh, GLP-1, okay. Yeah.
So one of the things that is really interesting that's coming out of all these cognitive psychiatry GWAS, like you rightly mentioned, these are like sodium channel genes coming out, calcium channel genes coming out. So absolutely, those would be kind of also linked to CVD, cognition, and psychiatric disorders. Now, where we start to look into drug repurposing analysis, for instance, so there are actually drugs that basically come out, cardiovascular disease drugs that are also coming out in those analyses. Now, the question is that going to rescue cognitive function or, at the same time be pleiotropic enough to have two functions? But I think that remains to be seen. Yes.
Thank you. So I'll move on to Nicolas. Unsurprisingly, I should put it up so everybody can read it. Trying to navigate the technology. All right, okay. Unsurprisingly, there was no difference in the number of structural variants between the three populations in Singapore, but are you able to compare with similar populations in other global biobanks, or have you done the comparisons, I guess? So like, I assume Indians in the UK biobank, do you expect to see similar things, or?
So that's not something we've done. And now that I read that question, I think, yeah, we could. We will. I don't expect much difference, but yeah.
Because I assume, I guess they're all migrant populations, and they might be different.
So yeah.
And India is big.
Yeah.
Right.
So sorry, no answer to that question, but yeah, there will be an answer provided.
Next year?
Yeah.
I think it just be a bit of a cautious because we know that NGS-based as we call it is definitely not perfect, and also very much platform dependent. Data depth dependent.
Exactly.
Will you compare all these cross different studies, or is this not sure if you see a difference? How can you be sure that difference is biological, or like you says is immigration or migration-based of thing, or because just reflecting the different technology platform, depths of data, all these things involved. This is, I think, it's very challenging to be able to answer the question. Yeah.
No, we'll bake that in, of course.
Yes,
I mean,
right?
no, but because also we have, we'll be in a position to bake that in. Yeah.
I wonder if the essence of this question is also related to kind of ancestral differences in terms of the genomic architecture in the population.
Great. Thank you. Well, maybe we'll look forward to the answer in a couple of years' time then. To JJ, was there any specific ways used to help resolve the acrocentric chromosomes?
Let me see. Okay. Any used to help resolve? Okay, yeah, well, figure out our DNA. Without figuring out our DNA part, you will never be able you can really resolve that chromosome. That is because very, very big influence RD on these astrocentric chromosomes. Yes, and hope we can resolve it. But at least there's still sequence missing there because RD made the whole thing very difficult to make sure they are complete or not complete. Yeah.
Thank you. I think this one must be for Max. Is there any study on whole genome sequencing findings and behavior problems, including sociopaths? And that's a loaded question.
That's a very specific question.
That's a very loaded question.
Yes. Well, there are kind of, not WGS, but I think microarray type of work that has gone into studying personality traits. Now, specific to sociopathy, I'm not too sure about that. I don't remember seeing any papers on sociopathy, but definitely, yes, there's a kind of genetic architecture underlying personality traits. The effect is not large, so it's not like, there are specific genes that will predispose personality traits.
Thank you. I guess defining sociopathy would have to have
Exactly. It would be hard.
The data sets would be a bit hard, I guess. I guess this one is probably to all of you. Given the Singapore's expertise in genomic medicine, are there any plans to share the knowledge and expertise through bioinformatics training to other Southeast Asian countries? I like this question.
Yes, I think a very good question. I think definitely it is. I would say not obligation or responsibility. We do need to share our knowledge and experience and capabilities. And how to organize this, should we down through the PRECISE, open to all the countries, or through any particular organization, universities? We should look into that. I'll feedback that question to PRECISE, and we can explore. At the GS, we do have a certain training course for our own students because many students come in as a very wet lab-based background, but they're now working on genomics. The data is very intensive. Then we have a very specific training course for our students to learn sufficient bioinformatics skill sets to be able to carry out their own project. Yeah, there are mechanisms for this. Yeah, good question. Yeah.
There may be possibilities for, a traveling expert, maybe if...
That one definitely need to ask PRECISE.
Yes. Because in some ways, being able to train in the local environment with the local, with the local infrastructure and limitation, I think is also important, right? Because I think it'd be one thing to be able to train in Singapore with everything is set up, and another thing as well to be able to train in the local environment within. So maybe there's ways we can figure how to do that.
Yes, I agree.
Absolutely. Yeah.
Max?
Well, I think the challenge is always too much data, too little analysts, and too little hands that's actually working on the data. But I think even in Singapore, we are having that kind of thing. Like myself, I'm learning from Nick how to run hail analysis. So that's Perfect, and I think there will be opportunities, certainly, as we come together as a network of biobanks in South Asia, groups in South Asia, to have that kind of opportunity.
And I think the momentum seems to be here now. I think there seems to be a lot more genomics initiatives around the region, and I think for some of the latecomers, it might be, we can kind of leapfrog a little bit and bypass some of, by learning from Singapore's experience. So that would be great. I have another question for Nicolas. You've mentioned how you've identified several structural variants in ACMG genes. Is there any intention to return results when doing a similar analysis in future studies?
So in the context of phase 1 of the National Precision Medicine Program, there was no mandate and no ability to return the results. As part of phase 2, some of the cohorts do have the intention to provide actionable returns of results. And of course, structural variation in ACMG secondary finding genes do qualify as being sort of cases for return of results. And so it will definitely be done, more. And then more precisely, the way it will be done is more actually we will help the genetic counselor basically do that particular part of the work, which is really, I'm equipped for identifying there is an SV.
Yes.
How to follow it up is, yeah.
Yeah, and we had a wonderful session before this.
Exactly, yes.
From the genetics counselors and the great role that they play. I have a question to all. All right. What are your opinions on linked reads or synthetic long reads kind of technology on assembling genomes, like the 10x Genomics or UST Chelsea?
Yes. Okay. I don't know. At least for humans, the day to use linked read is probably over. Okay, and the linked read, I have a limited experience. I only know what it can do, but I have a limited experience with all this. Is one thing they probably have advantage of other current true long read technology is their very, very low requirement in terms of input DNAs. They have very, very small volumes. For some very special situation, you just don't have a sufficient material to build a library for true long-read sequencing. This might help you a bit, yeah, at least to get some information out. That's my only feelings about that.
Yeah, I haven't had a chance to delve into this type of data, and I have a very sort of strong opinion one way or another.
Max?
I've not used these data before, so looking at JJ.
Thanks, yes. Jeju, then, for taking...
Hope can give, we are testing something of this. Yeah, hope we can share more in the future. Yeah.
There's a last question for, I think, JJ. Will you generate a new Indian de novo genome from new trio data or map it back to your existing assembly? And how do you plan to improve the challenging regions from the first Indian genome assembly?
Map it back to your existing assembly. We only have one assembly at the moment. I'm not sure when you generate the de novo from genomes. I'm not sure what you're referring to. We only have, maybe the person posed the question can help to clarify the question a little bit, and China region, like it says, RDNA. That's a very, very challenge. Yeah, I'm trying to clarify what does you mean by, we only have one genome at the moment, one sampling, put it this way.
Because towards the end of your presentation, you show that you will do more Indian genome, like 30 trios.
Yeah. Okay.
So will you use that data to map it back to your existing assembly, or will you generate new ones?
Okay, we're not really using mapping back because the current way is to, when you got the multiple genomes and to create a pan-genome reference, there's a way called a graph. to presenting this multiple genome, right? It's basically how you present them. Of course, you can present them just like a linear genome, one by one, and put it in the database. You can still do mapping against the linear genome, no problem. People do that, you know, like old days, we do the HLA coding, that's what we do. 1,000 different sequences right there, you just map your reads and do the HLA coding. They've done that before, but now people believe the graph is much more efficient way to presenting this pan genome. Yeah, this is our intention. You know, generate 100 genomes, build a graph to this. Given the point is there will be a 10,000 genome sequence by long read. There's additional variation will be there. Do we bring them in? Well, there is a plan to do that. Then the question is, when will be the end? There will always be another variation. Do you bring them all into the graph, right? So I don't know. I think you people still think about it, and at the beginning, I think it probably depend on what you're trying to do next with the graph. Define the question, then you will probably see how the graph should be constructed. Yep, thank you.
And on that, I've got the signal that we've run out of time. Thank you very much for the great questions and for the amazing answers.